All this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. Today we are going to talk about the T cell test that has been approved or actually authorized by FDA. This was authorized on March 5 or 6, I believe. So we are a little late in talking about it, but I think you would love the discussion. As usual, we have a beautiful gift here and we have a lots of illustrations. I was feeling a little tired because of the vaccine, so I took my sweet time to draw this out. So let's see, how did I do? So let's start a discussion. Hopefully everyone is doing good. The importance of this test is that with this test, we can understand better with more specificity that if someone was sick with COVID in the past. So the folks who have long hauler outcomes, they can actually figure out did they actually have COVID or not have COVID? Is there something else or this? That is one. Secondly, those folks who feel that they may have had COVID but have no way to tell and the antibody test come back negative, they may actually be able to do the T cell test and that would come back positive. So let's see why that would come back positive. So let's start. So again, another session in the Gifts for Humanity. So here I have a new um, color for the T cells. So T cell based COVID test. FDA authorized on the 5th March. The test is called T detect or T spot, and the company is Adaptive Biotechnologies. Let me very quickly show you the links. So if you wanted to follow those links and read them by yourself. So this is drbeen.com. This is FDA's authorization. So if you see here, March 5, 2021, and this is their authorization. Then this is the Adaptive Biotechnologies. That is what, if you read here, FDA authorizes Adaptive Biotechnologies T-Detect COVID test. So this is that company here, and this is a dedicated site for the test. This is something which is really weird, that newer companies do not put enough energy in putting their studies out in a more decent and marketable way. This is just an image of the study. No PDF, no link to the study, nothing, just here is the image. And as you increase this image in size, you can see that it becomes difficult to read. So anyways, I have the links for these things out there. And then this is another T cell test, but for research workers. So if you are a research worker and you are studying people's blood samples and you want to understand the T cells and you want to have a test that can give you profiles of the T cell, then this can be done. And this, I believe that since about a year, we have been talking about it amongst ourselves in the Cool Beans, that it would have been great to understand the T cell reactions as well, because that makes an important pathway in the immune response. And some people actually respond predominantly with the T cells compared to B cells. And so we should have some way of knowing the T cells as well. So here we have a test, and these tests used to exist more for researchers before. Now we have, for us common folks or individual people, we have the test as well. So these are the links. Let's go back to our illustrations. So here, the T cells are helper T cells, cytotoxic T cells, T regulatory cells, and other T cells as well. Helper T cells are also called CD4. So the CD4, cluster of designation 4, the cells that we talk about, all of our cells in our body, they have proteins on their surfaces. So imagine if the cell's surface is like an ocean, then there are tiny boats that are rowing in that ocean, or bigger ships that are there all the time. These proteins that are floating in the cell's membrane are called cluster of designations or protein markers. And many of these proteins are actually common across the cells, but there are some unique markers as well that are only found on a specific subset of the cell. So for example, T helper cells, and we'll discuss what is their function, we have talked about it in the past as well, they have CD4 markers on them. So if you can find a cell and you can identify a marker which is CD4 on them, then you should think that this is a this is a helper cell. On the other hand, if you see a CD8 marker, then you would say this is a cytotoxic cell, and so on. Now, what is the importance of this test? So let me go through this graph with you. So please follow along here. The basic problem is this. Are you right now infected? That is one. And number two, were you infected at some point? Two. Number three, were you asymptomatically infected? So all of those questions, and I have had those discussions many times with the cool bean as well, where somebody says, hey, you know what, in last November, I had a cold-like thing. I'm sure it was COVID, but I got my serology, I got my antibody test, and it came back negative. I still feel it was COVID. So in such scenarios, the T cell test would actually work. So let's see how would it work from a graphical point of view. So imagine that here somebody gets the infection. So I'm starting from the left side here. Somebody gets infected here. When they are infected, then the very first thing that starts happening in the body is that the virus starts increasing, replicating. So far, right in the very early time, the innate arm is slowly starting to respond. It takes about 24 hours to start responding. So initially, virus has free reign in our body. Nobody is responding to it. Then innate arm starts fighting with that. And as the innate arm starts fighting, the virus quantity ideally should start going down. So I'm talking about a normal, regular response where the virus would ultimately be defeated. So then what happens is that the innate arm starts beating up the virus, start killing it, start killing the infected cells, and the virus numbers start going down. Meanwhile, we also know, and I would go a little bit more in detail later on, meanwhile, what happens is that adaptive arm of the immune system starts becoming activated as well. It can take anywhere from 3-4 days to 17 days or more. And once the adaptive arm becomes active, in that arm, we have T cells, T helper cells, T cytotoxic cells, and B cells. So all of those become active. The B cells would start making antibodies. The T cells would start killing the cells that are infected. Now, when the B cell would start making antibody, then here is the graph for the antibodies, IgG and IgM. Normally, IgM is produced first, then IgG. However, they have observed that with the SARS-CoV-2, IgG and IgM are produced together. Still, IgM is a short-lived antibody. And so what happens is IgM would increase and then it would start reducing and it would go away soon. A couple of weeks later, IgM will disappear. IgG, on the other hand, is supposed to continue on for a couple of months to 3 months, and then that would go down as well. During this time, though, the memory cells will be formed, which will be memory helper cells, T helper cells, memory cytotoxic cells, memory B cells, and they will be put away in various parts of our tissues to be able to respond to this infection in the future. So now let's see how the T cell work or are useful. Let's start with this top line here, this line. In the beginning, let's say I get infected today. The PCR may or may not be positive. PCR is an antigen test. That means it picks up the pieces of RNA of the virus from the nasal and pharyngeal area, and then it amplifies them and tries to detect if the RNA is present. So that would only happen if the RNA is present in this area. So in the beginning of the infection, PCR test will be positive. Right in the very beginning, PCR would also not be positive, but in a day or so, it would become positive. Then, when the innate arm has started functioning, the virus has started growing as well. At that time, PCR will become positive. It has nothing to do with the innate arm, but in that phase where the immune's innate arm is working, PCR will be positive as well. Then, as we move forward and the virus is getting eliminated, PCR will become negative. So now if you say that, hey, do I have the infection? Maybe the PCR will be negative. If you say, hey, did I have the infection? PCR cannot tell you that. PCR would tell only if there is an active infection going on. Then the second test, serology, the antibody test, that is the most common test that we do today. So second line, check this line out with me. So what happens is, in the beginning, when the infection starts, the adaptive arm of the immune system has not yet become active. So there are no antibodies. So when there are no antibodies, we'll say serology is negative, meaning in the blood of the person, antibodies are absent. This is called serology. So serology is negative. Essentially, what we are saying is antibodies are not present. Then, when the adaptive arm starts working and the B cells become active, T cells become active, and we start having the antibodies, then of course the serology will become active. Now, this serology, I said active, positive. This serology would stay positive as long as the antibodies are being released. The cells that are releasing the antibodies, the B cell or plasma cell, eventually they are going to die, and some of them will be stashed away as memory cells, and they would stop working because there is no more infection. So, for example, once those cells have gone away and you did the antibody test, you would not get anything over there. So now you would once again start scratching your head to say, well, did I have that infection? Because there are no antibodies. So then comes the asymptomatic folks. There is another set of folks who mostly have asymptomatic disease. They may suspect that, hey, maybe I may have had the disease. My, let's say, family member became sick and I did not become sick. And when they do their serology, that comes back negative. When they do their antibody, that comes back negative. And this is a very common thing, even amongst you. If you see, many times people would say, my husband became sick, he was positive for COVID, I got my antibody test and I was negative. Very common. So that maybe these folks have responded with the cytotoxic T cell arm. And we have been wondering about that for the whole year. And I have always been saying that we should have T cell tests as well to complete the picture. So here we are. So now let's say this is the last line for the testing here. When the virus just arrived in the system, T cells will not be positive because they're part of adaptive arm. They would take some time to become activated. When the innate arm is responding, and by the way, innate arm is natural killer cells, neutrophils, macrophages, and many other proteins. When the innate arm is working, even at that time, T cells are not yet functional, and so T cell is negative. What I'm not covering here is reinfection. In the reinfection, these things will be different. I'm talking about somebody becoming sick for the first time with this virus or with any virus. Then when the adaptive arm has become active and the helper cells have started functioning, then the T cell will become positive. Helper cells, cytotoxic cells. They would continue to be positive. Then they would become memory T cells as well, and they would be present. So if we can identify the helper cells, and in the next slide, I would explain why helper cells in all cases will be present, why the T cells in all cases will be detectable. So T cell test would be more reliable than others. And let's see why is that. So here, just let's look at this little cute T cell. He's a memory T cell, sleeping but present, and we can always count on this little tiger, T for tiger as well, right, to give us a positive result. Let's see now why T cells would always be there even when the antibodies are not. So check this out. So I spent a little more time. This is when Dr. Mobeen is vaccinated and feeling a little tired. So here we have a dendritic cell, right? So we have antigen presenting cells, professional antigen presenting cells. These are dendritic cells, macrophages, and B cell. We can also have non-professional antigen presenting cells, which will be all nucleated cells. So I just took the antigen presenting cell here, professional. and it is presenting the antigen. That is, it has picked up a SARS-CoV-2 coronavirus, chopped it up into smaller pieces, and then it is presenting those smaller pieces on its MHC2 proteins. Here is a naive T cell. Naive T cell does not know yet that will it become a T helper 2 or T helper 1. It doesn't know that yet. It is naive. It is going to connect with the antigen presenting cell. First of all, it connects with the T cell receptor with the MHC2 of the antigen presenting cell, and they kind of shake hands. When they shake hand, so this is like they're hugging and kissing, so they have hugged yet. Now, if there is no kiss, then they are going to separate and not come close to each other again. So there is a co-stimulation needed. What that means is that this T cell will become energic, A-N-E-R-G-I-C. Energetic means it will not care for this antigen anymore if we very quickly not stimulate it with another signal. That is called a co-stimulation. So what happens is, as soon as this cell, the naive T cell, is connected through the T cell receptor, then we also need to do a co-stimulation immediately. And normally that is done through CD28 and B7. So these are also two proteins that they would also connect. So this is like, hugging and kissing and all that. So once that happens, then T cell will either become a T helper 2 or T helper 1. And we have done this discussion in the past that in the presence of interleukin 4 and absence of interleukin 12, the T cell, naive T cell, will become a T helper 2 cell. On the other hand, in the presence of interferon, so interleukin 12, I should have written somewhere over here, interleukin 12, which usually comes from these cells, the antigen presenting cells. In the presence of interleukin 12, usually the naive T cell will become T helper 1 cell. Good. Now look at this. This set of cells, this is an important statement now. This set of cells will always be present regardless of which path you eventually take. It is possible that T helper 2 is dominant and T helper 1 is less dominant, or T helper 1 is dominant and T helper 2 is recessive or less dominant. But this set, or this cell, or this cell, or this cell, they would for sure be present. So let's take this pathway, immunoglobulin pathway. So what will happen in this case is that T helper naive cell became active and became T helper 2 cell. This T helper 2 cell will become memory cells as well. These are the memory cells that they identify. So this becomes a memory cell. In this process also, the active cells would cause the B cells to become plasma cells, which would start making antibodies. Today, for blood test, we look at the antibodies. So that means what we are counting on to understand that somebody had COVID or not, we are counting on, number one, the pathway was taken this. Number two, the cells that became active were B cells. Number three, they are still making antibodies. B cells or plasma cells are not always making antibodies. They do their function, they die away, put some memory cells in the drawers, and then they go away. They don't make more antibodies. So that means if 3-4 months later, somebody who had COVID, they do the antibody test, it is not necessary that that test would come back positive. But if they looked at the memory T helper 2 cells, that would be positive. If they actually looked at some memory B cells, that would be positive as well. So anyways, going for memory B cell, because if you go for memory B cell, then you are in a very specific area of the pathway. But if you go for the memory T cells, then you are a little earlier. So antibodies present or not, T cell will be present. again, the specificity is in 90s. Now here, let's say that the T cell went to the T helper 1 pathway, that is in the presence of interleukin 12. The T cell went T helper 1 pathway. T helper 1 would release interleukin 2 that would convert other cytotoxic T cells to an active form. So if you see here, this is an active cytotoxic cell. I want to show it to you. I gave it a couple of weapons as well. So an active cytotoxic T cell is CD8 positive, and it has perforins. So this little spear-like thing is a perforin. It would use that to create perforations in the other cells, create punctures in the other cells. And it has granzymes, which are proteins that it would throw in the other cell through that perforation, through that hole it created with the spear. And then when the granzyme goes into the target cell, that cell would commit suicide and die. The granzymes activate caspases. Caspases cause tagging of the ubiquitin proteins and cause the cells to die. Generally, cytotoxic cells would kill the cells that are infected or that are cancerous or that are just not behaving correctly. So T cells will be there with their memory. So if we can find T cells, then we are great. So now the question is, how do we find them? So here is the method. So far, the takeaway is that the T cells are great little cells that, number one, help with the adaptive arm. Number two, they are memory cells as well. They become memory cells and we can identify them. They are our own little tigers. They are our own little Lufis. So let's see, how does this whole test work? And if you wanted to read this up, here on this test, if you go to the bottom of it, here, almost in the middle, in the FAQ, this is the paragraph that I'm going to illustrate for you. And for the medicos here, this is nothing new. This is detecting the antibodies through the fluorescence. So here is what is happening. You take the patient's blood, then from the blood, isolate the mononuclear cells. So in the blood, there are RBCs, they do not have a nucleus. There are platelets that do not have a nucleus. And then there are cells that have a nucleus. Mononuclear cells mean cells that have one nucleus. There are cells that are morphonuclear as well. So you choose those cells that are mononuclear. So you take them out. So these may have B cells, they may be T cells, they may be T helper cells, naive cells, and so on. So there may be many kind of cells, but you separate them. When you separate them, so you are picking them out of blood, so they're kind of dirty. They have blood all over them. So you shower them. You give them a shower and you clean them, you wash them. So once you have washed them, here these are the nice little neat cells. These are mononuclear cells. Then what you do is you count them. Why? For example, what we don't want is, let's say you take my blood sample, you take another person's blood sample. Let's say my blood sample already has 100 cells. Another person's blood sample has 10 cells. And then you try to work with them as a test, and I already have 100 cells, and you'll come back and say Mubin is positive. So what they do is they first count the cell in a specific part of the blood. Once they have counted, so this is my this is my depiction of Count Dracula. I laugh at my own diagrams. So Count Dracula would be called in, and that would count the cells. You would not take all of those cells, you would normalize them, meaning you would decide, you know what, we're going to take 10 cells. And this is just like with RT-PCR, you take a tiny amount of the RNA and then you do cycles, and cycle has a threshold. Similarly, here you take the cells and you count them, and you take a specific number of cells. The rest of the cells are thrown away. So then you put these cells in a plate. So it's not really one plate. There are multiple plates in which you put those cells. You give them food so they can continue to live and grow, and they are sitting there now in these plates. This actually, I just made it look like a plate, but originally in the lab, it is something like this. It's a board-like thing, and that has many wells in it, or there are plates in it, and then you stick the cells on that. Anyway, so here, we'll go with this plate. So you put them in a plate, you give them food, and then you put the antigen of interest on them. So imagine if this was my blood sample, you've taken my T cells out of it now, and now you are putting SARS-CoV-2 antigens on these T cells. These T cells, if they are there, if they are memory T cell, they are going to wake up and they're going to respond to it. Now the question is, can there be memory T cells that do not respond to SARS-CoV-2? Yes. Whenever we have some infection, we make memory cells and we stash them away. And they're circulating in the blood as well. They're just kind of sleeping and floating. According to them, they are in space, they're just floating in space. So it is not necessary that every helper T cell here or memory T cell would react to antigen or the antigen of your interest. You are interested that, hey, do we have any T cells here that would react to SARS-CoV-2? If so, then we have a T cell system that is aware of the SARS-CoV. But there may be helper T cells that are responding to, let's say, flu or influenza or Epstein-Barr or whatever. So those are all circulating and running around, floating around. So you put the antigen on them. If there are T cells of our interest, then what would happen is, what would happen is this. So see here, the red things are the antigens. So that is dead SARS-CoV-2 or pieces of SARS-CoV-2 that we put on the T cells. The blue ones, the dark blue ones, are the ones that have reacted. They are the ones who are saying, yeah, we know this thing. We're going to react to this thing. We saw this 3 months ago. And they start reacting to it. You might see that there are some blue cells here that are not reacting. They're just continuing to smile because they don't know this antigen. They're just like, whatever. But the others one have become active. And we know that when T cells become active, what do? They release cytokines. What kind of cytokines? We know that over here. See here that this T cell can, T helper 2 can do IL-4, 5, IL-10. T helper 1 can do IL-2. T helper 17 can do, Treg 17 can do IL-17 and 21 and so on. So they release interleukins or cytokines. Now what happens is, if we go back here, what will happen is that these cells have started secreting cytokines because they have seen the enemy. They have become active. Now we want to understand if the T cells have released cytokines. And how do we see that? Very, very interesting. What we do is this. We make an antibody against the cytokine of interest that we want to measure. Let's say maybe IL-2 or IL-4 or IL-5 or IL-10 and so on. Whichever cytokine we want. What are cytokines? Cytokines are little molecules, correct? So just like there are antibodies against other antigens, there can be antibodies that can be built to bind with these molecules. And you can buy these cytokines from chemical, medical companies. So now what we do is we take in, so this company's product, it has the antibodies that can bind with the cytokines of our interest. So if you put the antigen on these cells and they react and they produce the cytokine, then our job is to detect the cytokine. And for that, the first step is to have an antibody that binds with the cytokine. So if I continue here, now imagine in this diagram, start from the bottom here. In this diagram, at the bottom, this purplish thing is the cytokine. On this purple color cytokine, I have these black antibodies fixed on it. Why are they fixed? Because they can bind with this molecule. If T cells were not present, or if T cells were present but were not reactive to SARS-CoV-2, then this purple cytokine flood will not occur and these antibodies will not bind here. And if you would wash this, these antibodies will be washed away because they have nothing to hold on to. But here, because we have the cytokine, these antibodies have now bound to cytokine. Good. Now we want to know that, are they there? So what you do is you give them a wash, and now you want to see, are these there? To do that, you have one more antibody that is against this antibody, right? So second set of antibodies. But these antibodies have color labels on them. So what you do is you then, after washing it, you then put another antibody in this plate, in this well. If the previous antibody, the black one, was washed away because there was no cytokine, then this red antibody, when you put it, it will not bind to anything and there will be no color. On the other hand, if the SARS-CoV-2 was present, if the T cell produced the cytokine, if the cytokine was present, and if the bodies had bound to the cytokine, then these red ones will bind to these antibodies, and now the whole system is present. It is tagged with colors. Then what you do is you put more enzymes here that can now go and react with that color, and you can then detect those colors. These are called spots. So on this plate, where is that plate? That plate was somewhere over here. So here on this plate, if you had successful detection of antibodies, and some areas would show color, these are called color spots. This is why it is called a T-spot test. And those spots would tell you that there were T cells sitting in that area that are reacting to this antigen. And voila, we know that now the T cells are there. That is this technology. It's a beautiful little mechanism, and this mechanism is very commonly used in medicine, but I'm really, really loving it that we have this mechanism available for the SARS-CoV-2 as well. Now, if a person's antibody is not present, PCR is negative, but they still feel that I had COVID, then they can get the T cell test and they would know if really they had COVID or not. And this test is about $150 at this time. I do not know if the insurance has covered that or not. Still, if somebody wants to figure out what is their next therapeutic process, and if they're a long hauler, I think it is a little bit high at this time, but still it is worth doing. So this is the discussion for today. I hope you enjoyed it. What I'm going to do is I'm going to hang up now and then come back again for a quick chit chat, and then we will see further. If you have any questions, we can then talk about those in the chit chat. So please do me a favor. Please like, subscribe, and share. If you would not want to subscribe or share, then just like, and that is fine as well. And then some folks who became very upset with me with the vaccine I took, I think there have been 2,000 or 3,000 folks who unsubscribed yesterday. So I feel bad that they left. It's okay, they didn't want to be here anymore, but there is still things for them as well. Anyways, please like, subscribe, and share. At the least, like it. And if you would like to support this work, in the description there are 3 links. One is if you would like to buy me a coffee. The other one is if you would like to become a patron for this work. And the third one is if you just want to support this work. So thank you very much. I'm going to hang up, and I would see you in 5 minutes again. Bye-bye.